Hello everyone. I take immense pleasure in introducing the speaker of our next session, Dr. Venil Naranan Sumantran, Scientific Advisor, Dr. APJ Abdul Kalam Center of Excellence in Innovation and Entrepreneurship, a part of Dr. MGR Educational and Research Institute. Dr. Venil Sumantran has completed her PhD in the field of biology and biochemistry at Virginia Polytechnic Institute and State University, USA. She has also pursued 2 postdoctoral fellowships in University of Michigan. Dr. Venil is a senior researcher with 22 years of experience in top-ranked universities and research organizations in USA, Europe, and India. Her research expertise includes cancer biology and advanced biochemistry, molecular basis of diseases, biochemistry and instrumentation. She was principal investigator in USA NIH-funded research at University of Michigan. Her work experience spans across prestigious institutes like University of Michigan, Gothenburg University, Vision Research Foundation, and IIT Madras. Add to her credits, Dr. Venil Sumantran has published 43 papers in peer-reviewed journals, 3 book chapters with H-index of 20 and citations above 1500. She regularly reviews on BIRAC, SIBRI, and big grant proposals. Dr. Sumantran has developed in vitro assays to evaluate anti-arthritic activity of selected Ayurvedic drugs on human osteoarthritic cartilage. This work has produced 1 patent and 7 papers. At the behest of Dr. MGR.
You can start speaking now. We will all be going mute now.
Good afternoon, everyone. I hope you all can hear me. The talks so far have been very interesting. So for the past 10-15 years, I've been doing independent research on a low budget, and I want to talk about how to be the best researcher you can be. Not getting the next slide. So why do I think you have to do this? Because if you are the best researcher you can be, others will automatically want to collaborate with you. It's easier said than done, so I'm going to talk about what the obstacles may be in your path. I'll give 2 of the strategies for novel research that I have tried, and this really requires a small budget, so I hope it will help you. I'll also talk about why you should give your research an insurance policy. I'll explain that. And then finally, it's very tough to be a researcher. And maybe with the post-COVID-19, it may get even tougher for a while because funds may dry up. So you have to recharge your interest in research. You shouldn't give up. So let me begin with a Hindu newspaper article last year, which talked about the problem of copycat research. So there was a vice chancellor from a university in South India who said that the university faculty are urged to discourage irrelevant research, and in many state universities the research topics are often repeated. So we know about this, most of us. The PSA to the Prime Minister also added some points in this article by saying important basic questions in your chosen field must be tackled. Important applications must be developed. Now, obviously that's true, but I would like to dig a little deeper and see why we have these copycat research problems and what other problems we have. So these are some of the obstacles that I have faced: lack of funding, lack of trained manpower, lack of infrastructure, lack of time to read, develop good ideas for research. And let's be honest, there is also fear. I've come across this particularly in India. fear that someone may steal your research idea before you have time to work and publish it. And then last but not least, the fear of trying something new. And this has been my problem for many years. It's much easier to keep doing the same assay over and over again and publish papers. Trying something new requires guts and some risk-taking ability, and the earlier speakers have talked about this. So, having looked at these obstacles, what can I add that might help some of you? I'd like to talk about low budget research. I admit I don't have answers to all these obstacles, but by doing some low budget research, you can publish some quality papers. So that's what the next few slides will talk about. So one of the low risk strategies that I've used in the past 10 years is to identify a well curated database which has free data sets that can be analyzed with simple software or software that you can learn. Now remember, there may already be publications from this free data set that you choose. So read carefully, find new questions that these publications did not, and then reanalyze such data with your original questions and hypotheses to get a good paper. Now this is not a risky strategy, but it requires a lot of patience, willingness to learn, and a tenacity to keep at it. So let me give you an example of how this particular strategy worked for me and a group of MGRU students about 5-6 years ago. So we started with a free data set on gene expression in normal skin versus skin from melanoma cancer patients. Before getting into the data set, I had read melanoma literature, and it was exclusively on the gene markers for diagnosis and treatment. Now, as a biochemist, we all know that skin has a property of giving us a waterproof barrier. Why? Because it contains a lot of fat or lipid. We also knew from cancer research that cancers have abnormalities in fat metabolism. So our team at MGRU, we reanalyzed this data set for genes regulating fat metabolism. And we got some interesting results. We found specific fat metabolism genes and pathways were significantly activated in the melanoma skin compared to normal skin. And we were quite surprised that this was the first such report on melanoma worldwide. Why hadn't anybody else thought of looking at fat metabolism in skin? I'm not sure. I'm not a genius, we were not geniuses, but we asked an interesting novel question on a pre-existing free data set. So we can do it, anybody else can do it. That's my point. Another strategy that I have used, this was in Pune in the early 2000s, apply an inexpensive technique to a new research problem to get some novel data. So I was given the charge of a grant where we were looking at different Ayurvedic herbs for activity. This was a grant from CSIR and the work was done in Pune. And Amla and Ashwagandha are two herbs with very similar properties. And I asked the simple question, how can we distinguish between them at the cell level? I hope you can see the picture of the Amla fruit and the Ashwagandha plant on the right. So what we realized when we looked at the literature is that very few studies simply measured the effects of these two herbs on cell growth. I wanted to try that. I took two methods for measuring growth. One is the very popular MTT assay, which really doesn't measure growth. It measures cell survival, but it's a short-term assay. It's easy, reproducible. And the second assay was the colony formation assay, which is usually done in soft agar for cancer cells. we modified it for Chinese hamster ovary cells and we did it on plain tissue culture plastic. This assay really measures the division of one single cell into multiple cells, which then form a colony. So those of you who do bacterial work, you'll be quite familiar with a colony assay. So this colony formation assay can be done with mammalian cells too. And it takes longer, and it's important, very important I think, because it measures long-term growth. So we had an MTT assay for short-term growth and the colony assay for the long-term growth. Now let's look at the results we got. Fairly interesting. So here you see in 1A and 1B, 2 6-well plates. 0 simply means that no drug was added. Under 0 you see triplicate wells and fair number of colonies of the CHO cells. In 1B, on the right, we added 50 micrograms per ml of the amla extract. Not much difference in colony number. In 1A, we have 100 micrograms per ml of the extract. Maybe a slight difference, but we counted individual colonies, looked for statistically significant differences. There were none. So we were fairly confident. We repeated this experiment many times with different batches of amla, and we never found any inhibition of colony formation by amla, which is called Nellikai in South India and Aula in North India. Now, ashwagandha gave us very different results. So here you see 3 6-well plates. Again, 0 means no drug added. In all the 0 labeled wells, you can see a good number of colonies. And if you look at 2C, you can see 25 micrograms per ml, 2B, 50 micrograms per ml, and 2A, 100 micrograms per ml. The point is fairly clear that there's a dose-dependent inhibition of colony formation by the ashwagandha extract. So using the MTT assay, we never saw any difference between amla and ashwagandha. I've not shown you that data. But on the colony forming assay, we got completely different activities for these two herbs. And our Pune team's publication is one of the very few reports showing that ashwagandha and amla can have very different effects on long-term cell growth, not on short-term cell growth. So with these two examples, I've shown you how you can ask interesting novel questions and get good publications in decent journals without much of a budget. The third point I wanted to cover was the idea of an insurance policy for your research. That may sound a little strange. What I mean is, if you get a project from a funding agency, please do 2 to 3 types of independent experiments in parallel. Why? Because if one fails, chances are the other one will succeed and give you some good data. So again, going to my Pune experience, we had to test certain Ayurvedic grants for anti-arthritic activity. We actually did these experiments on cartilage from the arthritis patients who were undergoing surgery. So we would actually go during the surgery, collect the tissue, keep it sterile and work on it. We did 3 types of experiments on cartilage cells as well as the cartilage matrix. Here's just a picture to show you a piece of cartilage matrix, and you can see tracks, and within those tracks you can see the small cartilage cells which are called chondrocytes. So the first experiment was, can any Ayurvedic drug improve the survival of these cartilage cells from these arthritis patients? We got very variable results. The cartilage cells would quickly become fibroblastic and basically it became a big flop. The second experiment was, can any Ayurvedic drug slow down the loss of cartilage matrix from these samples? And we did get some successful data. We compared it with a positive control, glucosamine sulfate, and we could then get publishable, statistically significant results. The third approach, we didn't use any cartilage from the patients. What did we do? We actually bought 2 commercially available enzymes which can degrade cartilage matrix. One is hyaluronidase, the other is collagenase. We set up simple assays for these enzymes. And again, we found that some Ayurvedic drugs would inhibit these enzymes significantly, and we could get reproducible data and publish. So I've shown you the 3 types of experiments and how they could give us a patent and good research papers. If we had only done the first experiment on the cartilage cells, we would have nothing because it flopped badly. So this is the idea of the insurance policy. Please don't have one type of experiment. Please have 2 to 3 independent experiments which will give you a better chance of getting publishable data. At least this has been my experience. I'll go back for a minute. I should say that all of this work was done over the past 15 years. It's a very tough, frustrating journey. Many researchers would agree with me. And often I would face this problem where me and my team, we have a lot of activity, but where is the productivity? So it's really important for researchers to find ways to recharge themselves, just like we do recharging of all our mobile devices. You know, now you're staying at home in lockdown. Find ways to rekindle your interest in research, because it can be a lonely and an unrewarding career many times. Let's be honest about that. So the next few slides, I just want to tell you how I try to recharge myself. It's harder for me in a sense because I work alone, mainly from home, independently. I don't have people who I can regularly discuss with. So what do? I find that if I'm constantly doing computer work or administrative work, it definitely decreases motivation for research. I've seen that in some people around me, and when they get a break from all of that and they go back to the lab and spend hours a week, just a few hours a week doing experiments, they get refreshed. So this is one way to avoid the burnout that we face. And your students will definitely respect you a lot more. I remember my PhD advisor would constantly be purifying enzymes, working with equipment, and we had tremendous respect for him. He would come in on a Sunday morning and work. So please remember that stay in touch with your technical work for a few hours a week. The other thing is you have to force yourself to learn new things and apply them for your research, whether it's computer skills or a new assay or some new programming that you have to learn, or even entrepreneurship, innovation courses like we are doing in the Kalam Center. You have to upgrade yourself in different ways. That will enrich your research. Start interacting with other departments. Don't be in a silo by yourself. It's a small group discussion, a small journal club, whatever it is, start in a small way. It will definitely benefit you, your students, and it can lead to a very good collaboration sometimes. So another...
Excuse me, madam. Please in 5 minutes, please try to finish, madam.
Yeah, I'll be done.
Thank you. Sorry for the disturbance.
So the last point is about recharging your interest in research. It's very important to read, keep reading related and unrelated topics. The reading will sharpen your mind. You can ask good and relevant questions. Also review papers in your field or discuss them in journal clubs. It will give you ideas, solutions. You may even find a collaborator through that approach. So the main point is reading and reviewing is tremendously important for recharging yourself and getting good papers and grant proposals. So my summary is, these are some of the strategies that I think you need to be the best researcher you can be, and you can try them out. They will help you improve your publication record and then hopefully attract better collaborations. Thank you very much. This we can skip. Yeah, so stay positive and thank you.
Thank you. Thank you, madam. Excellent session. You have really made the session very awesome. Excellent presentation. We could see the excellent positive responses from YouTube viewers. More than, at present, more than 700 plus participants, and the like is exceeding 400 to 500. Thank you. I request Dr. Rama madam to post questions on
a lot of questions, but I'm going to just restrict myself to just one. How difficult was it for you to learn about microarray analysis when you did, and how difficult is it for you to convince students to learn the same?
I found it very difficult because I was scared of computers, statistics, and I still have that fear to some extent. I must say there was a professor and a student who helped me a lot. And then I kept working from home, making mistakes, nobody saw them. So that gave me some confidence. And frankly, it's been easier to teach the younger generation. I think they're much more interested in using online tools. So I find they're enthusiastic, but they also have a weakness in statistics. And so that's the only problem. They're enthusiastic and they are catching up much faster than I did.
Thank you, ma'am, for a wonderful talk. And all of us can see how passionate that you are about research, that you still continue with your research. Thank you, ma'am.
Thank you.